about algorithms. Algorithms in the whole life are usually boring, but sometimes necessary. And algorithm, in my case, has a bigger ambition to contribute for the evaluation of tumor markers for diagnosis and therapy monitoring in cancer diseases. So what is the background? Very shortly, in our project, biomarkers are used in daily clinical practice, particularly for the recurrence detection during the follow-up period of the cancer diseases. They are rarely used for the diagnostic purpose or therapy monitoring. Now, the aim of our study, of course, because not only me, was to propose algorithms for optimization of the diagnostic approach for the cancer diseases and for the choice of the optimal therapy and for monitoring of therapy effect based on serum levels of tumor markers. It means that we will speak about another family of tumor markers as before. So, our study is based on retrospective evaluation of 30,000 results of serum tumor markers from 2,000 patients, only for the region around Pilsen, and during the period of 10 years. And what cancers were chosen? We evaluated breast, colorectal, prostate cancer, and lung cancer. And the following markers were used. It means that they are from different families, and I will later explain you why. So I will answer the question you will ask me first. Why we decided these cancers and why these biomarkers? And the answer is very simple because it's a retrospective study. So we had information about this complete. So I will save your time. Then the next, results. So the results are concerning diagnostic algorithms in the tumor of unknown origin, differential diagnosis, early diagnosis, follow-up and disease course monitoring, therapy effect monitoring for surgery, adjuvant or palliative therapy, chemotherapy, and prognosis estimation. So, cancers of unknown origin. So we know that cancer is not a simple disease, so we usually meet patients suffering from different and combination of diseases. And we sometimes face the confusion of histology and immunohistochemistry. So the markers used here are named. So the first two markers, yes, are embryonic antigenes, cancer embryonic antigenes, and others are enzymes and also cytokinins. So we don't use them for primary diagnosis because of pure sensitivity and specificity, what we know, of course. Because in our inner check rules, when the biomarker can be used as prognostic, it must be, we have a very high sensitivity and specificity. And none of them used here can reach these levels. So now here we are with the first software which can help us to estimate what kind of diseases is it. If it is unknown time and location of primary tumor, when metastases are diagnosed or there is a high suspicion of cancer disease. So physician can add to this software, I have it in my laptop, how is it called, this small iPad, so it really exists, so you can find it, and it will help you to solve the problem where the metastasis is and primary tumor could be found. So in differential diagnosis, we can compare two illnesses, one malignant and one benign, and using combination of two biomarkers, we can reach 82% sensitivity and 95% specificity. But when we add another thymidine kinase, the sensitivity is 10% higher. The same is in another case, and we used for one or combination of three biomarkers, it is the same. result. Now, let me show you how we use this for optimization of early diagnosis in prostate cancer. So for us, this is prostatic-specific antigen, and the cutoff value is 10. So this value is index of health prostate and is calculated on another factors, and the cutoff value is 40. So what does it mean? It means that using these two parameters, we can save biopsy for these two categories of patients and also magnetic resonance. In the numbers, when we calculated 1,350 patients, for biopsies we saved 200, sorry. For magnetic resonance also 200, and for this combination 150 patients. So it's, yes, so it saved not only patient and the doctors, but also money. Disease follow-up monitoring. So this is a comparison of levels of CEA. In these two cases, they are, yes, oh, no, the left two columns are benign, and the three right are colorectal, breast, and liver metastasis. You can see the levels, the blue levels of CA before operation. And then you can see the levels of the first, second, and after the 30 days. What it means, in the left two columns, the levels of CNA were very low before during the whole period. And the cutoff level for CA is 5%. So you can see that here it is under the level, and the 30 days in all cancers, the levels were at under the cutoff value. Another, sensitivity of thymidine kinase in colorectal cancer, and this shows us the period prior clinical manifestation. Oh, this one is better. Thank you. Yes, thank you. So 9 months before, the levels of all these biomarkers were very low. And after 3 months, they were increasing a little bit. And you can see that when we combined 2 biomarkers or 3 biomarkers, the sensitivity and specificity was higher. So using the combination of this biomarker showed us better and more precisely the preclinical manifestation. This is the same in the breast cancer. So in 356 women, also you can see that we evaluated 3 biomarkers, so 3 separately, and the last column is together. So again, it shows better results prior the clinical manifestation. And this is the last example of liver cancer. Again, the same results. So the second software is called Corrector, and it will estimate the prognosis of certain diagnosis. So it will, when you ask this software, well, how it clinically evident metastasis develop in a case of a patient in a remission by the next checkup, it will help you to answer the risk of development of distant metastasis in the course of approximately next 3 months and their location. And the physician again can add additional information to make the prognosis and the answer from the software more precise. Now, therapy monitoring. This is a case of adjuvant chemotherapy in the cycle of 6 chemotherapy cycles. So the levels of thymidine kinases are not very high. Again, I mentioned that the cutoff is 10 in this case, so it's not. But after the chemotherapy, they grow up. It means that the chemotherapy was successful because, to make the long story short, there was blocked some synthesis around DNA, and the thymidine kinases had to grow up to support the normal life of the DNA and on the cell. So it is in the adjuvant chemotherapy. The next example is in palliative chemotherapy. So we started with the higher basic levels, but the shape of the line is the same. So also in the palliative chemotherapy, the thymidine kinase show the same results. Now in numbers, so this is successful palliative therapy, and you can see that these two additional biomarkers, which started in very high levels, were going down during, I don't know, 3 months, yes, rapidly. So for CA, as I mentioned, 10 is the right cutoff value, and in CA19 it is 40 or 50, so it's not as good result, but you can see that the tendency is very well. And this is example how when it doesn't work. So this is unfavorable prognosis for the patient in a palliative chemotherapy. And now here are results for C and CA19-9. You can see that the levels during only 2 months, but doesn't matter, are also the same. And so this therapy was unsuccessful. And now, conclusions. So we are in conclusions. So the optimal diagnostic algorithms were proposed for diagnosis and therapy monitoring for this mentioned 4 cancers. Selected cases reports demonstrated use of them and validity of them, and clinical and economical benefit of those proposed algorithms were evaluated. It means to save not only money, but also patient and the doctor. Next conclusion, a multidisciplinary approach based on these algorithms will enable to use the tumor markers for routine clinical practice much more effectively, not in the single case, but in the combination. Then perspectives for the methodology. It's mainly in multiparametric and multiplex assays with computer-assisted interpretation. We will use them in circulated tumor markers for the future, and they are also perspective in genomics, proteomics, and macroarrays. And as we mentioned here several times, personalized medicine in the approach, which it answers also these questions. For clinical perspective, the algorithms to individualization of target diagnosis and target therapy, correlation of biological activity of tumor with imaging techniques. Yes, this is new branch we are now focusing on, and incorporation of tumor markers into clinical and pharmacological trials. And this final goal is to prevent improvement in quality of life and better survival rates for our patients. Thank you very much. Hope I
So I know after the lunch everybody's a little sleepy, so I try to make things a little more interesting. This is look like a bad title is too complicated to understand. Let me pick the keywords. Oh, urinary. I'm very interested in urine. I think urine is probably even a better biomarker source than blood. Let me explain why. So that means a lot of people are looking for biomarker in not the optimal, may not be the right place. I don't know if that's too harsh, but let me explain why. I saw a few of presentations show the definition of biomarkers. Some says it's a protein or a metabolite. Some says it's a state of disease. But I think biomarker is like something that when you are healthy and you change to gradually to disease state, a lot of things changed. Some of the things changed are measurable. Some of them are not. Those measurable things are biomarker. So overall, biomarker is a change that changed from the healthy state to the disease state. So we are actually looking for a change. So that's why when we design a biomarker experiment, we always have at least 2 groups of samples. One is control, the one is diseased. One healthy, one diseased. Why? We always have to subtract, to compare, to show the difference. That difference is actually a change. So we are looking for a change. Where should we look for change? Are there a lot of changes in blood or more changes somewhere else? If you still remember the physiology textbook, the most important thing in physiology is the homeostasis of internal environment, which means the blood tends to be stable. Our cells are happy in a stable internal environment. We want the blood to be stable. We have all kinds of mechanisms, the liver, the kidney, the skin, the lung, to remove all the unwanted things in the blood to make the blood stable. So most of us are looking for changes in a place where changes are rarely happened, or happen soon and then disappear soon. So looking for biomarkers in blood is like playing hide and seek. You have to find the change before it being removed by liver or kidney. That makes things hard. But in urine, a lot of changes getting to urine. But also there are other changes getting to lung, get to liver, get to skin, but they're all easily to get contaminated by the feces, by the things in your stomach. Only urine. It's so pure, so clean. It's in the bladder for hours, just waiting for you to see what's happening in your body. So I'm interested in looking for biomarkers in urine. Actually, I talked about those last year. That's why I didn't prepare to do that. This year, the organizer asked me to give a workshop on preserving urines economically and easily. I was wondering, is anybody interested in urine at all? So before I do that workshop, I want to do an advertisement just like this. Urine is important for you too. And before, I always say there are more opportunities in urine, in biomarkers in urine. Today, I would also want to emphasize that the risk for not touching urine. I will give more evidences in the workshop, but actually, there are more and more evidence showing that for some of the biomarkers, they perform even better in urine. So you have like opportunities in urine, and you have risk in urine too. If you don't touch it. If somebody tests your biomarker in urine and it performs better, that's a big loss for you. That's why I think it's important to at least take a look at why we should look at urine. So since I talked about those last year, I didn't prepare to talk about that. This year, I would want to say, when we look at the urine in biomarker, the situation is very different from looking for biomarkers in blood. In blood, everything's stable. It's very hard to find a change. But in urine, it's very different. There's so many changes. It's so hard to find the one that associated with your disease. Let me show this. This is actually one of the pictures never get published, because this is the urinary proteins collected from healthy individuals in my laboratory. Graduate students, they're all healthy, young, but the patterns look different. That's probably one of the reasons not many people working on urine. One of the pioneers, Dr. Moses, just gave us presentation yesterday, showed beautiful result many years ago, but not many people working on urine. not following the suit. I was wondering why. I guess a lot of people stop here. When they see there's so many differences in healthy urine, we don't see a smooth baseline. How can we find biomarkers in this kind of environment? But I see it differently. If biomarker is a change, and there's a lot of change, there's a lot of opportunities. That also means they're all healthy, there's changes. That means there are some physiological changes can be reflected in urine. If physiological conditions can be reflected in urine, pathological conditions should be reflected even earlier and more sensitively. So that's why I think urine is a better place to go. But there's a lot of problems in looking for. Today, I want to talk about one problem is when we always have two groups of people, healthy control and patients. We try to stratify with gender, with like same group of male, same number of male here, same number of male there, and age, we all stratified. There's always one problem we can't do it. That's this medication. Ethically, we cannot stop the medication of the patient just for the sake of biomarker discovery. But we can't give same medication to the healthy control either. So when we do the profile of these two groups, there's healthy and disease. At the same time, there's closely associated another factor, which is no medication and medication. That make the balance change. So we have to assume that medication may not change the biomarker. We hope. If the medication do not change the biomarker, the whole scheme looks fine. But if that's true, I think biomarker, the medicine will definitely change the biomarker. Let's think about it. Biomarker is you're from healthy to disease. Medicine is change you from the disease to the healthy. It's supposed to remove some of the biomarkers that disease causes. And at the same time, it probably produce other changes too. So medicine helps the patient to change from disease state to the normal state, and medication must change biomarkers. Not possible. When you are completely healthy, you will see those biomarkers should be gone. That's the effect of medication. So here, not many people study like biomarkers of drugs, of medications. So I wanted to show some of the examples that we did in the laboratory, some of the diuretic, anesthetic, and anticoagulant to see if they really have effects on urine. I try to make it faster in those details. Different kind of diuretics. Basically, it's a self-control. You take the urine and give the animal medicine and see what happened to the urine afterwards. That's the result of diuretics. You see all the rats in the disease in the medicine-treated group changed, and some of those proteins that changed were considered biomarker before. That means when they talk about those biomarkers, I don't know if they really noticed that some of the medications may cause that biomarker to change too. That's the same things happen too. And they are some homologs were considered biomarkers too, and they are all easily affected by diuretics. And these two are the anesthetics that we used to test. Similar thing, the similar result. Some of them are considered candidate biomarkers, some of them are not. And those are the validations. I don't want to go too much into the detail. And the third one is actually we tested for if urine is a better biomarker source. We try to change the function of the blood, see if we can see more difference in blood or more difference in urine. One of the fundamental function of the blood is coagulation. Very basic. No coagulation, people die. So when we change with anticoagulant to change the function of blood, guess what will happen more sensitively in blood or in urine? Actually, with the same standard, same approach, everything's the same, we can see more changes in urine and more changes in urine and less change in blood. At my title, I said, in urinary biomarker study, we should take care of the medication, the drugs. But actually, even for people who work on the biomarkers in blood, you should take care about the drug's effect too. Even though there's only a small number of proteins change, but they may actually change your result. Let me make an example. Some of the people with the stent in the body, they may use anticoagulant. But when you try to study the effect of stent or the atherosclerosis, you may not notice all the people with stent, they may have some kind of anticoagulant. And those healthy control, you can never give them anticoagulant. So that marker may come from two sources. One is drug, one is the real effect. So one way is we study the drug, the effect of drug in animal, and when we study the biomarker, we can subtract that one. So I want to skip that summary too. This is another important thing that I think is very useful for studying urinary biomarkers. Just like I said, for studying biomarkers in blood, the problem is to find change. There are so many proteins, and the body only allows little change. You cannot have too much change because people will die if you have too much change in blood. But in urine, you have less protein. You can tolerate a lot of changes. The problem is to find changes. It is to find association, the change and the disease association. How to do it? For clinical studies, it's very hard to control. And what we eat, what we drink, if we do exercise or not, they all change urinary proteins. It's very hard to have exactly two groups of people, diseased and healthy people, exactly the same. Another reason is medication. It's very hard. But I think we can minimize the factors by using animal models. So if we find the clue from the animal models and then validate in human sample, maybe it's much easier to find biomarker this way. That's actually that I prepared. So I've talked about earlier in the talk. Biomarker is a change. Urine accumulated a lot of change is a better biomarker source. That's why we want to save it. Save it easily, economically, at room temperature for a long time. So I want to just leave a little time for questions.
The title of my talk is Clinical Biomarkers in Drug Development, Quantitative PCR-Based Fit-for-Purpose Assay Qualification. Now, this is a collaborative effort, and the work is carried out at Biocon Bristol Myers Squibb Research and Development Center, Syngene International Limited, located at Bangalore, India. So biomarkers have a lot of potential uses throughout the drug development process. And right from target selection to lead identification, biomarkers have many potential uses, like it can be used for target identification, drug screening, candidate selection, lead characterization. In the preclinical validation phase, the biomarkers can be used to assess the mechanism of action of the drug, to establish the in vivo proof of concept, to determine the toxicity and the potency of the drug that is being tested. And all through from phase 1 clinical trial to the product launching, biomarkers have again many different uses, which I will summarize in my next slide. But before going to the next slide, the evolution of biomarkers actually ideally should parallel the drug discovery pipeline. So the minute a target is selected, one should have a biomarker discovered along with it, as in the morning half also I think Sid alluded to this particular fact. Once a biomarker is discovered, it should be confirmed and an appropriate assay should be developed. Following that, the biomarker should be validated not just in the preclinical mode but also in the clinical setup to establish the role of biomarkers in disease pathogenesis, and all the way up to the regulatory approval and the clinical adoption of the biomarker. So what role does it play in the clinical trials? So I've just divided into two different sections, one the early phase and the next is the late phase. So the first in human single ascending dose or multiple ascending dose proof of concept kind of studies in the early phase clinical trials, the biomarkers play a very important role in terms of providing supporting data which will help us in giving an early go, no-go decision. So it is a very important tool in making this kind of decisions. And it also provides support for proof of concept studies. So there are various different types of biomarkers that can be utilized for this kind of purposes. The biomarkers can also be used for dose selection, again something that was alluded to in the morning half, to go into the next phase of the trial, that is from escalation to the expansion phase. In the late phase, I will just summarize and say that the biomarkers are utilized to enhance the overall quality and the scientific strength of regulatory dossiers. And they are also used as companion diagnostics for various different disease indications. So what would be an ideal clinical biomarker? It should be, the expression should be significantly increased in the related disease condition. It should be shown to correlate with an interested outcome or disease progression. The biomarker should be readily quantifiable in accessible biological fluids, as we have been hearing now from since yesterday. It could be blood, serum, plasma, urine, or it could be cells and tissues, or even whole body. For example, whole body imaging techniques are available in the market like X-ray or CT scan or PET MRI, which are used very routinely in medical practice, for example, to look at the response of tumors to therapy. And last but not the least, and very important, is the biomarker should be measurable in a very consistent manner, and preferably it should be quick and economical. And as we have again heard a lot of times that in the last one day that the biomarkers, there are various different technology platforms which are available in the market to assess the biomarkers. So these could be genomics-based, proteomics-based, or cell-based assays, and I've just listed a few. Today's talk will be focused on quantitative PCR. So depending on which core technology platform is being used, the early clinical and translational research department in BBRC, that is the Biocon Bristol Myers Squibb Research and Development Center, we have 3 different subgroups that carry out assay development, assay qualification, and then using these qualified assays to analyze clinical samples to come to the proof of mechanism concept. And the different disease areas that we look at are from the therapeutic areas of oncology and immuno-oncology, and fibrosis is a new addition to this portfolio. Okay, so all the 3 subgroups use something that is called as fit-for-purpose biomarker assays. So what is this fit-for-purpose biomarker assay? The biomarker data must be reliable and accurate, and we should know what is the intended purpose of that particular assay. So the rigor of the validation method depends on what kind of assay is chosen and what is the use of that particular assay. Is it for discovery purpose, or is it for proof of mechanism or proof of concept kind of purpose, or is it a prognostic or predictive kind of purpose? So based on that, the rigor keeps increasing for the assay development and the assay qualification, as the biomarker data is used for increasingly advanced clinical studies or for making very critical business decisions. Hence, the analytical validation requirements are specific to the stage of the drug development. Also, we need to consider the intended use of the biomarker data and consideration to the regulatory requirements associated with that use, plus it should be practical and an iterative approach. So this slide basically gives an overview of the clinical biomarker assays that are carried out in our laboratory. So what is the intended use of the fit-for-purpose assay? To assess the pharmacodynamic or PD biomarker changes due to drug treatment in peripheral blood and tumor samples during phase 1 clinical trials. So starting with the selection of biomarkers, followed by assay development, and then a fit-for-purpose assay qualification is done. This qualified assay is then used to analyze clinical samples to arrive at proof of mechanism of drug action. So in the next few slides, I will be talking more about the assay development and the assay qualification part of it, and then show one example wherein this has been used to derive the proof of mechanism of the drug action. All right, so as I already mentioned, today's talk is focused on quantitative PCR as a technology platform. And we all know that there are various advantages of using quantitative PCR as the method of choice. It's rapid, reproducible, robust, sensitive. It has a broad dynamic range. It's affordable, and it is quantitative in nature. However, there also exists many challenges with it. One is the lack of technical standardization from lab to lab. So often it happens that although we come across many papers which report results, we often find that we cannot reproduce that result because of lack of technical details that is given or reported in the paper, and hence the results often differ. In our kind of setup, wherein a huge number of clinical samples are analyzed, what we have found is that there is also a lack of skilled operators for high throughput setup to carry out this kind of analysis. Hence, it becomes really important to consider an appropriate assay design as well as stringent and robust standard operating procedure development. So the most common assay that we carry out in our laboratory is quantification of the levels of mRNA expression, and it's a relative quantification that we do because these are all clinical trial samples, so drug treatment, without treatment, and then various time points of the treatment. So the sample quality, which is in this particular case, it's going to be RNA. So the sample quality and the source of sample becomes extremely important for us because any variability in the sample or the quality of the sample affects the results in the end. So it should be robust enough that the assay is consistent over a period of time. So the sample source, usually it is blood, tumor biopsies, bone marrow, etc. The homogeneity of the sample depending on what is the sample type. In case of blood, if it is collected in Paxgen tubes, it should be mixed properly. The tumor should be collected in RNA later so that the integrity is preserved and it should not get degraded. Sample storage, the time between collection and storage, these are all worldwide clinical trials that we are talking about. So each site of the central laboratory which handles this sample collection, they should have standard SOPs to collect the samples and store the samples at an appropriate temperature. The shipment procedure should also be standard. The freeze-thaw cycles, which affects the RNA stability and the integrity. Then once the samples are received in our labs, the RNA extraction procedure also matters a lot because it could carry the contaminants and that could bias the recovery. There are other factors like DNase-1 treatment. There should be no genomic DNA present because that could affect the PCR efficiencies, and also the yield, which includes the purity and the integrity of the sample. And what we follow in our lab is a very stringent method of ensuring that the RNA quality is maintained. So the 260/280 ratio should be between 1.7 to 2.2, and the RQI, which essentially reflects the integrity of the RNA measured by different instruments, and that should be equal to or more than 7. However, the criteria is little relaxed in case of tumor biopsies because those are very precious samples, and it's just that we can get a baseline sample and a post-dose sample. So there are no replicates, and hence even if the sample is found to be a little bit degraded, we still go ahead and do the analysis. So this slide now highlights the different steps in the qPCR-based assay development process that we follow generally. The design input is based on the program requirements, the biomarker expression, that is normal versus the diseased condition, and depending on the number of biomarkers that we need to analyze, if it is a small number, we usually do a single gene analysis, and if the number of biomarkers to be analyzed is more than 12, then we use something called Tachman low density array based cards to analyze more than 12 samples or more than 12 different genes. And coming to designing the primer and probes, we usually stick to pre-designed, inventoried, and custom-designed Tachman assays from Life Technologies. And we try to keep this standard because it may happen that our laboratory develops the assay and qualifies the assay, but the assay could be transferred to another third vendor somewhere else in the world. So they need to be able to source the same primer probes for their use. And as usual, the primers should be across exon boundaries, preferably, to avoid genomic DNA contamination problem. The next step is selecting something called quality control samples. So these are essentially, they act as the references for high, mid, and low level expressions for the selected target and endogenous control genes or the reference genes. And again, we try to select QC samples which are commercially bought so that again, the same RNA sample can be bought by any other third party. And in case the commercially bought RNA does not express the genes to the desired level, in that case, we also make it in-house using either tumor cell lines or other cell lines which have positive expression. And ideally, what this represents is that the QC sample representing high, mid, and low should be differing by 2 to 3 delta CT values. So delta CT in this case is the gene of interest expression level, which in case of qPCR is expressed as CT or cycle threshold, minus the endogenous control CT value. So that's the delta CT. So this should be the difference to maintain a particular range. So continuing with the assay development steps, the next is the selection of the endogenous control genes. So typically we choose 1 to 3 endogenous control genes because not a single endogenous control gene may or may not live up to our expectations. So it is better to screen more right at the beginning. And the criteria is that the expression should be stable within initial target population for the PD biomarkers. And it should also be stable in response to the compound or the drug. So here is one such data represented here, wherein this is before compound treatment, after the compound treatment, and this is in the normal blood. So the expression level, as you can see, is almost similar in all the 3 cases. The next is to determine the PCR efficiency and the linear dynamic range. So ideally, the PCR efficiency should be between 100 and plus minus 15% is tolerated. The dynamic range should cover more than or equal to 3 log values of the amount that we use. And this just shows one such typical cDNA dilution curve analysis wherein the PCR efficiency for 4 different genes are calculated, and they are all within the acceptable range. And since, as I mentioned, these are all relative quantifications that we do, so the relative quantification is the genes are normalized to the endogenous control genes. So the relative efficiency is measured with respect to the endogenous control. So they should have a slope of less than 0.2 in case of single gene, and it's little relaxed in case of the array cards because there are more number of genes to be fitted in the criteria. So the slope is relaxed to 0.25. So once the assay is developed, we come to precision studies to qualify the assay. So what we typically do is do an intra-run precision test by carrying 3 repeat measurements per run, wherein the standard deviation should be less than or equal to 0.5. And for tilde, again, the criteria is relaxed a little bit to 0.6. When we do the measurement of RNA levels, it is a 2-step process. From the RNA, we first convert it into a cDNA using a reverse transcriptase reaction, and then that is then put into the quantitative PCR, the real-time PCR reaction. So it is very important to set the range using the different RT reactions, that is the reverse transcriptase reactions. So 3 independent RT reactions are done, and the standard deviation again should be less than 0.7. For the tilde, the standard deviation is decided on a case-by-case basis. And here is a typical example of 3 different inter-RT runs here. wherein the delta city values are depicted and the standard deviation was found to be only 0.22. So that's really tight intra-run variation here. And then for each operator or analyst who does this kind of assay, the QC range is defined as average delta CT plus minus 2 standard deviation. So whenever an operator changes over, the minimum qualification that they have to do is at least carry out the inter-RT and establish their own QC range. In addition, we also do consider site-to-site variation, so lab A versus lab B, for example. In case the instrument changes, we also do instrument-to-instrument variation and operator or analyst-to-analyst variation to design the precision studies. So here is a graph shown for a 48 gene panel tilde card, wherein 2 different endogenous control gene performance was measured. And as you can see, between 2 different laboratories, X versus Y, the standard deviation for most of the genes are within 0.6 except for few. Okay, all right, yeah. So essentially minimal assay variations were observed in all the key genes for the precision studies, and hence the assay is taken to be validated for the gene expression analysis. The other 2 tables here show manual versus automation. So in case manual pipetting versus JNS liquid handling system, for example. So again, we have to establish the range and make sure that the standard deviations are not outside the range. And as I already mentioned, even for operator to operator. So why are these qualified assays so important? Because they serve as a very powerful tool in clinical trials. This is just an example of a clinical trial that has been ongoing in our laboratory for the past 5 years now. The data is for a QC high and a QC low sample here for 4 different genes. 2 are endogenous controls and 2 are PD biomarkers. And as you can see, of the 33 runs, it has been consistently showing the expression levels over a period of 4 years now. So this essentially strengthens the reliability on these assays, and delivering these kind of high quality data enables in decision making early on in the clinical trials. The next slide is live oncology. Thank you. This is an oncology trial that has been ongoing in our laboratory, and these are 2 different PD biomarkers, and this shows the different time points, and this is the mean of the relative expression levels of the PD biomarkers. And over a period of time, this shows the dosing cycle of the drug treatment. So as you can see, 50% inhibition is taken to be very significant. So over a period of time, it shows consistent downregulation of the PD biomarker upon treatment with the inhibitor, indicating drug hitting the target, hence establishing proof of mechanism. So with this, I would like to summarize that qPCR is an important technology for rapid, sensitive, and economical measurement of clinical biomarkers. There are many key factors that influence this kind of assay, and as biomarkers become integrated into drug development and clinical trials, the fit for purpose assay qualification becomes increasingly important to make decisions early on in the clinical trials, and also to save money, which is because the clinical trials are extremely expensive. So these are the people I would like to thank, Michael and Mohan, for sponsoring this particular talk. Zenhao Chi is my collaborator from BMS. Sharat Pujari is my team member who has contributed significantly in the last 5 years to this particular work, and all other past and present members of my team. And thank you for your attention.
Maybe I should start over with a little more detail. Why should we save urine before we do it step by step? Why do we have to save urine if it's hardly used? So we have to find a reason why. I claim urine is a better biomarker source than blood. As I said, there's all the other definitions, but I believe this definition probably express or emphasize changes enough. It's a measurable change associated with disease. The change can be protein, can be metabolized, can be microRNA, can be heart rate, can be body temperature, but anything change that associates with the disease is a biomarker. if you play a word game to cross off all the less important one, the most important word left is the change. So biomarker by nature is change. And change is hard to be found in blood because in blood there's homeostasis mechanism, which means in blood all the changes are minimized or removed, so there's less change and less biomarker, especially in the early stage when the homeostatic mechanism is still working. But people may ask, we have so many biomarkers in blood. Why do you say there's not enough? Those biomarkers can only be established when the homeostatic mechanism is out of balance, is out of control. If you have a little high blood glucose at the early stage, you won't see a high level of blood glucose. You have a diabetes, it's because all your homeostatic mechanism is all used up. You still can't control the blood glucose, and then you see it increase. That's why I believe the markers in the blood is hardly to be early and sensitive one. It may be relatively late stage and when the homeostatic mechanism is running out. So I always try to give an example like we use a lot of vinegars in Chinese cuisine. Like we do dumplings, we use a lot of vinegars. When you drink, let's make it more extreme, you drink vinegars and you drink a lot. What will happen in blood and in urine? Do you see the pH change in urine first or in blood first? And I believe before the blood running out of its capacity, we will see the urine change first rather than the blood. So that's why I believe if this mechanism holds well, we will see more changes more sensitively in urine rather than blood. And in blood, let me give it another example. Let's suppose there is no homeostatic mechanisms. This is against all the physiological textbook. If there's no homeostatic mechanism, you eat something in and they stay in blood. You take a medicine, you take a medication, you take a sleeping pill. What will happen? You will sleep forever. There's no mechanism to remove it. But actually, the homeostatic mechanism is so strong, no matter whatever you take, no matter the body ever seen it or not, they will always find ways to remove it, no matter what medicine. Like proteins can only stay in blood for a few minutes. The half-life is only a few minutes if they are not antibody. Antibody stay longer. But all the other protein drugs are really hard to stay. They are removed very quickly. And another example is like doping in sports. I don't see them really take any sample of blood samples. They can always find a trace amount of changes in urine, no matter what drug you used. But in urine, the good thing is it's accumulate change. But what do they remove? They remove all the useful part for your body. If they're useful, we keep them stay in the blood. We only remove all those that we don't want, and put them in the urine. So in the urine, it accumulates all the changes, and at the same time, keep the useful proteins in blood remain in blood. I borrowed these slides from the web just to show how the homeostatic mechanism works. There's lung, skin, liver, and kidney. If you drink alcohol, you can remove them from the lung, and a lot of medicines are removed by P450 from liver, and some other things like urea even from skin, and a lot of them from kidney. People can imagine why do we do biomarker study in blood? Because blood connected to all the organs that we wanted to see. We wanted to observe. But urine is not. But actually, urine contains all the unwanted one in the blood. In the blood, it's like a passage, even though it's connected to all the organs. It's the passage. This is the reservoir. This is the place where all the changes stay. That's why I believe maybe we can have like mutations or what kind of for another thousands of years, but I don't see we can have another pouch with such a clean liquid directly connected to blood, which we can use for biomarker research. Here are some of the examples. This is one of, they're not from my laboratory. I hope this can be more objective. And this is from Texas, University of Texas Medical Center. This is one of the examples that with the same people, the biomarker were tested in serum and in urine. This is angiostatin. In blood, they're not performing that well. That's chronic kidney disease, SLE, the lupus, and that's their performance in urine. You see much better p-value compared to the blood. The reason, just in the afternoon, I talk about why even the people who work on the blood, they should at least think about urine, the possibility of urine. I did a search on PubMed. When we have biomarker and blood, serum, and plasma, there's 300,000 papers since there's word biomarker, 300,000. Less than 10% with the word urine in it. But actually, the research is done in urine, even smaller, maybe 1,000 paper directly done, not many, which means a lot of people have done this part. They tested if they work in the blood, but they never tested if they work in urine. That give other people a big loophole. Let's imagine you work very hard to find a biomarker in blood. Let's sell the house and file the patent and form a company. What if somebody tests your biomarker in urine, and just like this one, and found that that biomarker works even better in urine? All your effort is down the drain. That's a separate intellectual property. But most people stop here. That's why I say there's a lot of opportunities. If you work on serum biomarker, mend the fence. Or if somebody do it for you, that's a big loss. But I encourage everybody who do biomarkers, at least if you really get addicted to blood, take a look at urine. That's another example. That's 4 biomarkers. They were done in urine, plasma, urine, plasma, urine, plasma, urine, plasma, all at different stages of cancer, I guess. This is done in Seattle and people in Germany. You see all the positive rate numbers. You compare the urine one and the plasma one in each of the conditions. This is the only exception. In plasma, it performs better than urine. In all the other categories, this is almost useless, but this one is 100%. There's a lot of, this is done 2009. There's a lot of evidence that for the same biomarker, it is possible that in urine it performs even better than in blood. I don't want to say it happens in all the biomarkers, but at least in some of them. This is actually from Dr. Moses' lab, but Dr. Moses is not here, who worked on the MMPs, and she found that's what she said, demonstrate that urinary MPs in determining the human cancer both inside and outside of the urinary tract. So even outside of urinary tract. At the beginning, I thought urine is only good for biomarker discovery of urinary system. But Actually, there's even more. This is from UK, a biomarker from COPD, lung disease, congestive obstructive pulmonary disease. That's the biomarker performance in urine, U-desmosine. You see the value in urine, the range is 6 to 10. In patients, it's 14 to 22. There's no overlap. 8 to 10, 14 to 22. But if you see the same biomarker in blood, there is an overlap. The highest value in healthy is 0.23. The lowest in patients is 0.21. When there is an overlap, it's really hard to use clinically. So that's a lung disease. I think this is also from Dr. Moses' laboratory, MMP9 and MMP2 in the brain tumor. This is the thing, actually, we thought about how far can it go, the condition be reflected in urine. Brain is the furthest place that we can think about. We were trying to design an experiment that happened the disease happened in brain to see what happened in urine. And one of the postdocs found that Dr. Moses have already done one of the example for us. And that's the one she showed yesterday. Before operation, after operation, this before operation, after operation. The biomarker is gone. There are also other brain ischemia. You see in serum, hardly differentiate. But in urine, there are two different proteins. The difference can be big. And that's the one that I talked about in the previous session. We use anticoagulant to change the function of blood to see where can we detect them more sensitively. In urine, there's more protein changed. In plasma, there's less. Urine, there's more. In blood, there's less. That's exactly the same strategy, same analysis, same machine, and same cutoff, same threshold. Everything's the same. Urine is very informative. When I talk to some of the physicians, that they don't believe there's proteins in urine. They believe if there's protein in urine, people are sick. There's some problem in the kidney. But actually, I have studied that for over 10 years. We haven't seen any person, any healthy person, without urinary proteins in their urine. They all have proteins. And this is unpublished data in the laboratory in my students. And he found that it's already 4,000 proteins if you really analyze very carefully. You really separate them well in the LC, and you can have over 6,000 proteins. And there's thousands of metabolites. I believe there's database in Canada, and there are microRNAs too. And there's actually very low background. And as I compare, I showed there's a lot of protein in blood, and the change is allowed, not that big. But the urine, we never seen any people died of too many proteins in urine or too many acidics in urine. People will never die, no matter what happened in urine. There's, the tolerance is big, and the background is low, and you can have enormous change. That's the 4,000 protein, the 6,000 protein they analyzed. Interestingly, those are the proteins that enriched. It's not very specific. It's not the protein that only specific to brain. It's enriched in brain. It means most of the time, most of those proteins stay in brain, but you can see it in urine. Kidney is not even the first one. There's more brain-enriched proteins and more liver-enriched proteins in urine than in kidney. And there's also other major organs. That means if we analyze the urine really well, urine can probably reflect the changes relatively specifically of esophagus, skin, testis, small intestine, duodenum, and those kind of organs, you probably will see changes relatively specifically. That's the one that I try to show. There's, when you do PubMed search, most people working on the at blood and not many people working in urine, and the percentage is very low. That's even just the presence of the word urine. That one, actually, I talked about in healthy people, there's a lot of change, which means if urine can reflect physiological changes, it probably can reflect early pathophysiological changes more sensitively. It can probably reflect pharmacological changes very sensitively too, because all those are probably bigger cause than the physiological change. But there's disadvantage of using urine as biomarker. There's all big variations. That's one of the reasons most people stop using it. And so we need a lot of case to validate. We need patients who drink Coke, Diet Pepsi, beer, alcohol. We need all those kind to see those biomarkers are not affected by the beef you eat, by the cheese you eat. So we need a lot of samples. But the problem is urine sample is very diluted and protein can be degraded, even you put them in -80. I can find a paper who showed if you stay longer enough, as long as it's in the aqueous condition, enzyme not completely lose their activity, even in -80. They're still working slowly. So eventually, you will have some kind of degradation. So the way we try to save urine is to remove the most costly liquid, the water. The water is not that informative as long as we measure the volume. We don't need the water we don't need to keep the water anymore. The water is most costly to save. So the way to remove water is to put it on, go through a filter. This filter is not by pore size, it's by absorption of protein, like nitrocellulose or PVDF. The protein stick to the membrane, and the water part is gone. Here is the membrane. Here is the membrane. When the protein is on the membrane, the way to keep them dormant is by let them dry. When you make it dry, all the enzymes stop working. Nothing can work without water. So the way is to make it dry and seal it in a vacuum bag to keep it dry all the time. As long as you keep it dry, it can stay at room temperature for very, very long. We haven't determined how long it stays yet. We tried for 3 months, no change in the SDS gel. In the mass spec, the proteins are all the same. And think about milk powder. Even without vacuum, they stay. How long do they stay? They stay very long. And for this kind of cheese, you can put it in the drawer or put it on the bookshelf. It doesn't need freezing temperature anymore. Save a lot of money by keeping it. Actually, before, I didn't realize this can be important. I thought about this idea originally for the lab to collect urine samples from very rural area. We can't bring refrigerator there and bring it back. So I said, what about you stick the protein on the membrane and then bring the membrane back? So we dump the water over there so we can work on it. Until one day, I believe this can be even more important than what I said, what I thought about. If we do it even in the hospital, that works even better. So this is a word that I quote from the Mayo Clinic. They are very proud of Henry Plummer, who is the architect of modern hospital. And hard as it to believe today, a single concept developed by Henry at the beginning of the 20th century changed the face of medicine. what's that idea, change the medicine? The idea is centralize the medical record. I guess before that, we all share the same medical record. We probably don't have a separate number. We all have, we probably on the same sheet. It's very hard to see the disease progression. And from this concept, everybody, we have a separate medical record. We can really see how the disease progress. That's the basis of medical research for this century of medicine. And it's now they developed very well. The medical record is paperless, filmless, chartless, but unfortunately, the medical record, which is so important for medicine, is still biological sampleless. We don't have any biological sample in our medical record. We don't have a hair, we don't have a nail, we don't even have a tooth. Even those, they are like non-invasive and harmless, we still don't have. Why? Because they are not informative. I think urine is informative enough and easier to save enough. Just about 1 or 2 months ago, I went to a biobanking meeting, and the organizer asked 3 or 4 of us at the podium to say what's the future of biobanking. I thought about, I think ideally we should preserve easily acquired, non-invasive, informative samples long-term at room temperature. This is about ideal. I think that URIMAN serves the purpose. It can be easily acquired, non-invasive, very informative, and can stay long-term at room temperature without any special storage condition, and it's cheap. So overall, I think urine smells good. And if you try it in the lab, work on it. Don't drink it. And that's the people who work on it, and the funding from Ministry of Science and Technology of China. And I hope I can save more time for questions. And the graduate student in the lab, Fan Shuang, will show us a video that they made how exactly that urine is urinary proteins and nucleic acids are saved on the membrane. And I don't know, should we have question and answers first, or later to show the? Oh, sure.
When you get the membrane, you let me wash that, because a lot of salts may be in the membrane as well. Then you get the membrane to the water, and the salt will come back and create more protein.
Most of the salt should stay with the liquid. I believe, yes.
But you don't wash it.
Yeah, we don't do it. Yeah. Okay, maybe Fan Shuang will come to show us the video.
Preserve urinary proteins on the nitrocellular membranes. Here is human urine sample. And first spin down to remove cells and cell debris. Then add phosphate buffer solution to make all urine samples to the same pH value. And assemble the vacuum system. The vacuum suction bottle is installed and connected to the vacuum pump. Full sheets of wetted filter papers are placed onto the bottle. And one nitrocellular membrane is placed onto the filter papers, avoiding the generation of bubbles. The urine collecting bottle is installed and sealed. Load with urine sample. Turn on the vacuum pump. Let the solution passing through nitrocellulose dropwise by adjusting the vacuum pressure. After the urinary proteins are adsorbed onto the nitrocellulose, the protein-bound membrane is taken down. Dry the membrane at room temperature or blow dry with hair dryer. The dried membrane is sealed in vacuum bag using a kitchen vacuum sealer. At last, write down the sample information. And if we change nitrocellular membrane into positively charged nylon membrane, urinary nucleic acids can also be preserved. That's all. Thank you.
For aging and disease. And to do that, I'll be explaining my stem cell theory of aging and disease. They say it's good to open with a joke, so this ad from the UK for telomere testing is my joke. It's really kind of a sick joke, actually. Do you remember the movie Blade Runner where the android knew up to the very moment when he was going to be dead? Well, telomere testing is decidedly not that kind of thing. Companies that recommend telomere testing have an interest in oversimplifying things, but the fact is, it's a little more complicated than that. There is no app that can tell you your exact date of death, but there is an app that can help you live forever. That app is called telomerase, and it's contained in every single stem cell in your body. That's what we'll be discussing at the Thomas Noguchi Sculpture Garden, right over there behind that building. But first, let's go to the Rose Hill Cemetery, where my father's father, and my mother's mother are all laid to rest. It's funny, they always try and get me to buy a little pied-à-terre over there, just like I rent a space in this parking garage. But what if this park won't need to park there at Rose Hill Cemetery until the 24th century? I'll explain. We're here at the beautiful Rose Hill Cemetery to illustrate a point. The loved ones of the people interred here probably thought they all died of something different. But consider these articles, thousands of articles, all showing that telomere dysfunction is related to the diseases that took them. In this cemetery, my mother's mother, my father's father, and my own father was laid to rest in 2006 after he lost his battle with brain cancer. So, how long do I really think I can live? Well, here's a chart of my telomere lengths since 2007. As you can plainly see, they've only gotten longer, so I may already be immortal. But even if I were to stop taking telomerase activators and age at the normal rate, this suggests I may have 250 years to live. Given that fact, I think I'll hold off on buying a plot for now. Back in 2006, my father was about to succumb to his brain cancer. Dad was a general practitioner, and he served in Saskatchewan for many years. He knew what was happening, but neither of us as physicians understood why, the why of cancer. When I Googled or looked up why people get old and sick, really only one explanation made sense to me, and that holds true to this day. That is a telomere theory of aging. As you all know, the ends of the chromosomes have telomeres on them. These telomeres shorten every time a cell divides. Now, when the cell thinks that they're too short, it can't tell the difference between that and a clean break, say from a X-ray. So what it does is it heals ends together. That causes double chromosomes like trapeze artists connected at the hands. And the next time cell division occurs, you'll get the centromeres and the telomeres pulled apart, but there'll be tears and mutations in the cell. Enough of these tears and mutations leads to the cell recognizing that it's damaged, and it causes the cell to go into crisis or apoptosis. Now, if I was to take a cell from your body and put it in a dish, it would only replicate 50 to 70 times because of this end replication or this replicative senescence problem. That's the Hayflick limit. In contrast, we know that cells over the course of a lifetime only age about 50 to 100 base pairs a year. How is that possible? Well, that's because our stem cells are constantly regenerating their telomeres, just like this stem is constantly regenerating itself. These dead pine needles have no telomerase activity, whereas the further you go to the trunk and the root, the more stem activity there is, the more telomerase activity is. And that's why stem cells are immortal, and that's what leads us to my theory of aging and disease. We're standing in the sculpture garden by Osamu Noguchi called California Scenario. In 1936, a young Osamu Noguchi asked Buckminster Fuller to explain E equals mc squared, and Buckminster Fuller telegraphed back this interesting message. It's funny, Einstein spent his entire life trying to find a theory to unify everything, or a grand unified theory that would explain how electromagnetism, gravitation, and the strong and weak nuclear forces were all united. Well, we're here to explain my theory, my grand unified theory of aging and disease. So let's do that right now. Axiom number 1, telomeres always shorten when the cells replicate. It's like water always flows downhill. Axiom 2, non-stem cells do not activate telomerase. Therefore, they do not live forever. These dead pine needles were not meant to live forever. Axiom number 3, a daughter cell can only be as healthy as its most recent mother, and it can only be as young as its most recent mother. This statue is called Spirit of the Lima Bean, and it should be obvious that a lima bean can only be as healthy as the lima plant from which it came. Axiom number 4, stem cells use telomerase to restore the length of their telomeres, thereby staying young. Just like somewhere under this pyramid, there's a pump that needs to restore the water back up to the origin over there. So, my hypothesis is that telomere shortening in your stem cells and the accumulated damage is what causes aging. It follows that if we can prevent or reverse this telomere damage, then we can prevent and reverse aging itself. We're standing right in front of the El Toro Y, where the 5 and the 405 freeways meet in Southern California. It's 26 lanes wide, and the reason we're here is because when you look at a normogram for telomere lengths from any study or any lab, they're going to be 26 lanes wide. In other words, you cannot predict someone's chronological age based on their telomere age. Having said that, telomere measurements are very precise and they're very reproducible. So it's a great way to personally benchmark for individuals. Now, if you consider this chart from the Kaiser study, 100,000 patients, what you can clearly see is that the trend is not your friend. There are three methods to measuring telomeres. Some would say they're like comparing apples to oranges, but I prefer to compare them to different types of orange juice. Let me explain. The first is the gold standard Southern blot, where you take the DNA, you cut it up with restriction enzymes, and then run it on a gel electrophoresis. It's very precise, but it's very labor intensive, and hence not available for public use. Just like this juice, the gold standard Southern blot comes with a little extra, be it pulp or calcium. So in that measurement, you get about 4,000 base pairs of sub-telomeric measurement added on. The most commonly available test is quantitative PCR, which would be sort of like juice from concentrate. Why? It's easy to make, you don't need fresh cells, and it's very affordable. The problem with quantitative PCR is that it has a little bit of inter-user variability, and so you have to always check and make sure that the tests don't vary too much. Said that probably about 7% variance exists between the same blood sample run on different samples of quantitative PCR. The last method is the fresh squeeze method. That's high throughput quantitative FISH. With FISH, or fluorescent in situ hybridization, you actually need live cells and you have to paint the DNA, then measure the amount of light that is painted on those telomeres. I prefer this method because it gives you a direct measurement of all the ends, and in one case, you can actually get a percentage of critically shortened or damaged telomeres. We're here at the Santa Ana Zoo to talk about animal models of aging. Many people believe that since laboratory mice have long telomeres and they still age, this proves that telomeres are not involved with aging. In fact, mice are inbred mutants, and they have freakishly long telomeres in the lab. Also, they can't respond to oxidative stress in the same way that humans can, and therefore have very high rates of genetic mutation. Now, this is a gibbon behind me, and our closest relative, as we know, is a chimpanzee. Chimpanzees in captivity conditions like this only live on average to their teens when you include infant mortality. Why is that? Well, perhaps it's related to the stress of being in captivity. Dr. Jane Goodall, famous primatologist, says that in nature, chimps can live into their 50s and 60s. What's the difference? Well, maybe chimpanzees with their twice the telomere length, yet twice the telomere erosion, could live as long as we do if they only had Amazon.com, better public health and clean water, and hot yoga classes. We don't know. I believe that AIDS is a form of aging, and aging is a form of AIDS. They are both each other's cause and each other's shadow. For example, chronic infection throughout our lives with CMV and other herpes viruses grinds down the system. It's that overuse, overcopying, and replicative senescence that causes us to lose our immune system and to age. Why do men like Jack LaLanne, who are physically fit, or Nelson Mandela, who are spiritually advanced, why do they die of the common cold? It's because they ran out of blank keys. Their naive T cells were used up. Consider individuals with trisomy 21 or Down syndrome. They age at 3 times the normal rate because they have 3 times faster telomere attrition due to their dysfunctional T cells. I'd like to take this moment to talk about the idea that telomerase activation causes cancer, as some of the experts in this field may have suggested, despite recommending telomerase activation by natural things, such as meditation. I believe the greatest contribution is the landmark study in the Journal of the American Medical Association in 2010 by Willett et al. What they showed was that there was a dose-dependent relationship between telomere shortening and both the incidence and mortality from all causes of cancer. Another interesting study out of Northwestern University by Howe et al. In 2015 showed that this is happening in 2 phases. They looked at men from the Veterans Affairs and looked at their charts. They found that telomere erosion happened quickly up until about 3 to 4 years before the clinical cancer manifested. This is in keeping with my stem cell theory of aging disease. Rapid telomere shortening allows the ends to cause fusion, which then cause mutation in that cell line. The crisis stage is when the stem cell is damaged and it's negotiating with itself, its neighbors, and the police or the immune system as to whether it will manifest as a clinical cancer. I believe that we are destined to get every single cancer if we can survive long enough. It's like having a car for 1,000,000 miles. You don't expect to have any of the original parts. Same goes for clinical cancer. Now, I subscribe to a cancer stem cell theory, which is that cancer does not, as we were perhaps taught in medical school, arise from differentiated run-of-the-mill cells. Rather, it primarily affects stem cells. In other words, you have an already immortalized stem cell, which then mutates. It doesn't have to learn how to immortalize through telomerase. It retains that capacity. It's like saying Jimmy Olsen doesn't learn how to fly, he's Jimmy Olsen. But Clark Kent, already being immortal and flying, can become Bizarro Superman. So, now we understand that the excessive copying, the replicative senescence happening in your stem cells is what's causing the cancer. And all those risk factors that we associated epidemiologically speaking are really just proxies for accelerated cell copying without enough time to regrow the telomeres. For example, smoking, coal mining in the lungs. The lungs have to turn over too fast, the stem cells can't keep up. Drinking in the esophagus, in the liver, same thing. Unopposed estrogen for the breasts and for the uterus. Unopposed ovulation, overuse of the ovaries for ovarian cancer, and excessive stimulation of the prostate by high testosterone levels. One of my biggest pet peeves in telomere research, and there are over 15,000 articles in the literature, is that most of them assume that blood cells and their telomere lengths will represent aging in other organs. The good studies study the organ of interest. For example, if I were to measure the tire pressure just because it's easy, will that tell me what's going on with the carburetor, the brakes, the electrical? Of course not. If this car came from Colorado, the underbody might be shot from road salt. If that car came from San Francisco, the brakes might be shot. Same thing with our body parts. Study the organ of interest and its telomere lengths. So that's really most of what I wanted to tell you about telomeres as a biomarker of aging and disease. Now it's up to you if you want to do the science that will help validate this grand unified theory. They used to say that there's 8 million stories in the naked city. Well, what if there's only just one? My name is Dr. Ed Park, and it's been my great privilege to present to you at this 2015 Toronto Biomarkers Conference. Thank you for your attention, and I hope with your efforts we can keep parked in places like this and not the other place. I wish that you and the telomeres in your stem cells will live long and prosper.